So, hello everyone. I'm Luciana, and it's my pleasure to be here talking to you today. I'm going to talk about our instruments at GenScript, more specifically about the AmMag Quatro for plasmid purification. You might wonder where my accent comes from. I'm originally from Brazil, and I'm now the head of the field application scientist at GenScript. And I actually did after my PhD in Brazil, I did working with structural biology in protein biochemistry. When I moved to US, I switched to molecular biology and cell biology at the University of Michigan, where I studied GPCRs and other cellular receptors in cancer, studying cell proliferation migration. So it's my pleasure to be here and to start this. So I'm going to cover today, I'll give you a little bit of an introduction about GenScript, a little background about plasmid purification, and the GenScript solution for plasmid purification. I'm going to talk a little bit about selected application data that we have, and then the Q&A. So we have been around for a while, since 2002, when it was first founded in Piscataway, GenScript. But we can see a lot of benchmarks there. The only thing that I want to call your attention that you might know GenScript because of our services, gene synthesis, protein, antibody. But we are another department from the GenScript that is instruments and catalog products. So let's give you a little background on the history of plasmid. So in early the 50s was when the plasmid was first discovered. And then on the 60s was when the plasmid was actually found to be circular. And then later on the 70s, we actually discovered many of the methodologies to clone plasmid DNA. And only later on the 80s, we have identified many methodologies to extract and purify plasmid DNA from the bacteria. So if you are talking about the methodology itself, we can separate this methodology in two parts. The first part is the cell lysate, so you have to open up the bacteria, and then after that, you do by alkaline lysis. That's a very effective to remove proteins, cellular debris, and genomic DNA. So after you have your lysate, you are going to follow up with the purification. There are a lot of methods out there to purify plasmid, and I'm going to list a couple that everybody must know. That is ethanol isopropanol precipitation, cesium chloride ultracentrifugation, and then boom method that is based on the silica membrane kits. And the most common one, that is the anion exchange columns. That these are all the kits in the market out there is with the anion exchange columns. However, our methodology is based on a magnet silica beads. So the traditional way to purify plasmid DNA, you know that is very time consuming and have several steps. So the step is including the resuspension of the pellet, the lysis, the neutralization, centrifugations, binding, washes, and then finally the eluting your plasmid DNA from the column. So this involves several steps of centrifugation and passing through the columns that can clog, and then you don't get your plasmid DNA for all your samples at the time you want to. So what we designed for you, it is a fully automated instrument for plasmid purification. So, and this was only possible because of the magnet beads technology, so the silica beads. We synthesize the core, the iron core, the ferrite, and then we cold coat with the silica. The silica has the property to bind to plasmid DNAs. And these actually solve most of the problems in terms of to scale up. Also, the ability of the beads to achieve a solid phase reversible immobilization is a very key feature because the magnetic beads can reversibly bind DNA under dehydrating conditions. So when using a strong magnet, it can be immobilized, as you can see in the figure, and throughout multiple steps of washes and manipulation, you can wash because your plasmid DNA is bound to the beads, so it is immobilized there. So this makes it possible to put everything, all this methodology that includes the magnet beads methodology in one instrument and to automate the whole process at once. So here you can see on top is our Maxi prep instrument called AmMag Quatro 1400. For short, we call it Maxi, Quatro Maxi. This is a fully automated plasmid DNA instrument for Maxi preps. And the beauty of these instruments is that you can have one controller connected to up to 4 modules. So if you, for the Maxi prep instrument, you can process up to 24 samples. So each module can process up to 6 samples in approximately 2 hours. But then after we launched this Maxi prep instrument, like it was 2 years ago, we now launched the mini prep instrument, the one below that we call Quatro Mini or 1100. So this system is very similar in terms of the methodology that we implemented into the first system. And the beauty of this Quatro series is that you can connect up to 4 modules, and doesn't matter if it is max or mini, so you can mix and match up to 4 modules. So the mini prep instrument is also fully automated, and you can, if you have all 4 modules connected, you can purify 192 samples in approximately 90 minutes. So the GenScript solution is not only the automated instrument, but is also the pre-filled consumables. So we design specifically all the reagents that has the formulations to achieve you the best yield and purity of your plasmid DNA. So, and also, we also provide the pre-programmed verified protocols. So as you can see in a closer image here, inside of the instrument, this is the mini prep instrument. You can see all the spots for each specific component of the kits that you provide. That is the 4-well plate. That is, you just need to grow your cultures inside of the 48-well plate. And then after centrifuge, this is the input inside of the instrument. Then you have all the reagent containers there, the final collection plate for your purified plasmid DNA, the tips, slots, and you can see also the 8-channel pipette arm over there that can move in the X and Y positions. So the total runtime is about 9 minutes, as I mentioned, and you're going to get a transfection-grade plasmid DNA. So the overall workflow works this way. So you grow overnight cultures inside of a 48-well plate. And then after that, you have to centrifuge. And basically, this is all you have to do that is not automated. Then you have to put your 48-well plated inside of the instrument and with all the consumables and hit start. When you hit start, the first step is going to be the media removal. And then followed by the resuspension, lysis, and neutralization of your lysate. After that, you have a step that we called pre-clarification. That's the incubation of the pre-clear beads. The pre-clear beads are the beads that are going to bind to cellular debris, proteins. And after that, we have one important step, that is the incubation for the binding beads. The binding beads are the beads that bind to your plasmid DNA. And after that, we have several washing steps followed by the dilution step, where you have your plasmid DNA delivered to the collection plate, the final plate. Here is one example of one of the software protocols. That is the interface, the software interface. As you can see, the beauty of the system that you can change several parameters inside of each protocol, such as the volume of your resuspension buffer, binding beads, pre-clear beads. But most importantly, you also can change, if you want your plasmid to be more concentrated, you can elute in less buffer. Here is a quick video to illustrate what I just talked about. I'm not sure if the sound is not allowed, but I hope you can see that this is the controller. This is the protocol interface, and then setting up your run, and then you basically click start, and you can walk away, and the instrument is going to do your mini prep for you. So I was telling about the precise of the pipetting. So the pipetting accuracy is 97% with a coefficient of variance of 3%. And what you get is a transfection grade plasmid DNA. By the end, here is the all Quatro series, the Maxi and Mini, and the future instrument that is coming to include in the series that is the Quatro instrument to purify proteins, also magnet beads based. So finally, I select a couple of slides to show you the application data. So here, this is one study showing we select a couple of high COP plasmids and low COP plasmids. So here we see the yield of a high COP plasmid being about 40 micrograms total plasmid, and the low COP plasmid is about 20 micrograms. The 2,6/280, that is telling us about protein contamination, all above the threshold that would be above 1.8. And the 2,6/230 ratio, that is going to tell you about contamination of other reagents, all above the threshold that is about 2. So another study that we conducted in our lab was we would like to understand better. These are bacteria growing plates. Which kind of media give us the best yield? So as you can see in the plot, we analyzed TB, Plasmid Plus, and LB, and we increase the volume of the culture from 1 to 2.2. So if you see this data, you can tell that TB is way richer than LB, and so Plasmid Plus as well. And what we can see that is the highest yield that we obtained was growing for about 2 mL of LB in a 48-well plate. The same thing, we analyzed the ratios and all above the threshold for this set of assays. And interestingly, we also analyzed the endotoxin levels for these two conditions, the LB and TB. As you can see in the plot, that LB actually has a very low endotoxin levels for all the samples, all below 0.1 EU per microgram. In comparison to TB that had greater endotoxin levels. So this is a good example where you can actually choose the media depending on your application if you are concerned about the endotoxin levels in your samples. Here, we also conduct a lot of diagnostic digest assays and analysis of the supercoil out of our instrument, the plasmids that was processed through our instrument. So as the data show, they all pass the diagnostic digestion of double digested by EcoR1 and SML1, and the supercoil ratio all above 95%, with genomic DNA very negligible. So these studies to show how uniform inside of a 48-well plate we have all these plasmids. This is from all from pcDNA 3.1. And you can see the concentration is very uniform, all about 400 nanograms per microliter, so the yield. And also the analysis of the endotoxin levels on these samples was very uniform, all below 0.1 U per microgram. This was conducted using LB. We also compared our purified plasmid DNA with the competitors. As you can see, we used 3 different plasmids to perform this comparison. In a high COP plasmid, for example, the PTT5, we see a very similar yield with low endotoxin levels. The PET28 is a low COP plasmid. We see a little bit more yield comparing to the other vendors, the other competitors, with low endotoxin levels. And for the back plasmid, we also see a little bit greater yield compared to the competitors. So finally, I wanted to show one of the studies that you conduct at GenScript that was you used the plasmids comparing to the competitors as well to do transient transfections in HEC293 cells. And we did it by flow cytometry comparing the EGFP positive cells. As you can see in the plot, that is very comparable. So the plasmids, they are able to be transfected efficiently compared to the other methods of purification. Finally, as you know, we are GenScript under cataloged products and instruments. We have not only the Quatro series, but we also have the other instruments. So the first one you can see that there is the mini prep instrument that we were going to show in the booth. Now we brought the instrument here. And then we also have the Maxi Prep. This was launched 2 years ago. We didn't bring this though. And then we have the protein purification called SA+, also magnet beads based. And we also have a couple of small devices for Western blotting. We have gels, and particularly I really like this e-stain. That is an instrument to automate your comassin staining. So you have your gel stained and de-stained in less than 10 minutes. We also have the cell isolation and activation beads and instrument, the CytoSync. I would like also to invite you to visit our booth. The number is 301 to see the instrument, to talk with us. We also offer to do virtual demos or demos with the instrument. You also, if you choose to do a virtual demo, you can send the samples to us. We process, we actually show the instrument how it works, and we send the plasmids back to you for your own validations. GenScript is also sponsoring the reception today, so come see us and take a drink and talk to us. We also have a poster section, 3 poster sections at the end of that room. So thank you very much, and I'm open to any questions if you have so.